Thank you very much. My name is Johannes Klueppel. I'm coming from the University of Freiburg in Germany, and I'm a PhD student in the EcoSense Collaborative Research Center. This research center they're coming environmental scientists and engineers together to investigate how forests and trees, and especially the photosynthesis, suffers from the dry summers because of climate change. And in the beginning, I would like you to ask a small question, because my research is very interdisciplinary. Can you mention the 5 main elements of photosynthesis process? So we have water from the soil, we have CO2 from the air, and we have, of course, light as an energy source. And what we get is glucose for the tree to build up and oxygen for the humans to breathe. And during my talk today, I would concentrate on the light as an energy source, how we measure it for photosynthesis, and why our calibration method for multispectral sensors improves the state of the art. So obviously, leaves are green because they reflect green light. But they absorb blue and red light, and it's now mentioned in photons, and you will also see later why. And you know, a blue photon has more energy than a red photon. And because I'm an engineer and not a biologist, and I think you as well, I feel responsible to briefly introduce the photosynthesis process to understand why we counting photons. So the photosynthesis happens in leaves and of a tree on the surface in the so-called photosystem. And when now a photon irradiates on the photosystem, it is absorbed by the chlorophyll, which is here in green dots, and then it bounces from chlorophyll to chlorophyll until it triggers an electron movement. And this is now a very important thing you have to remember. So we have one photon, one absorbed photon, so it doesn't matter if blue or red, is responsible for one electron movement. So it doesn't matter, so it's not very wavelength specific, so it's independent. And the environmental scientists call this radiation photosynthetically active radiation, which is basically something in the, so from 400, the number of photons from 400 to 700 nanometer, which is the visible light. But this is also used by the trees to make photosynthesis. And in a classical way, it is measured with broadband photodiodes. They have a filter from 400 to 700 nanometer. And in some versions of these commercial products, they have also a special weighting filter to weight the response of the photodiode inversely proportional to the energy of a photon, so that afterwards in your photodiode, it doesn't matter if there's a blue or a red photon, you get always the same photocurrent. And then you can calibrate for the number of photons. But these kind of sensors, they have a big disadvantage. It's only one channel, you measure over the whole spectrum, and light is never the same. So if you measure under a tree, you will have a red or green light. If the sun is covered by clouds, you have red or blue light, and if you have a clear day, you have a red or red light in your spectrum. And for agriculture also, it's very important that you have artificial sources which then have also completely different spectra. And therefore, researchers had the idea to measure this kind of value with multispectral sensors. So multispectral sensors, they come as an array most of the time, and each channel here has a distinct bandpass filter on top, and is responsible to a bandwidth from 20 to 25 nanometer. And luckily, this sensor here is also from 400 to 700 nanometer, and we have now the opportunity to weight each channel separately to also obtain this inversely proportional response to the energy of photons. So, and now I'm coming to the calibration. So was it previously done? So researchers, they just took these commercial broadband sensors next to the multispectral sensor and calibrated it under either artificial or sunlight. Then you have on the one side, you have your reference value. On the other side, you get the readings F of all your channels. You do a regression and you get the calibration coefficients. And you can then calculate your reference power value. Works perfectly. But as I told you before, sunlight is never the same. So the spectra of sunlight is never the same. Imagine you calibrate under a rather blue environment. You will read your power reference value. You will have readings of your multispectral sensor in the blue channels here. You do a regression. You get regression coefficients for your blue channels. Your red channels, green channels, they get a coefficient of roughly 0. You apply your coefficients then to the readings of your sensor, and you will perfectly calculate your reference power value. But imagine you use the same calibrated sensor now in a different spectra environment. It's a rather red spectra here. Of course, it's an extreme case. This will never be in nature like this, but just to make it clear. You again have power reference, but now you measure in the red channels of your multispectral sensor. Your coefficients are calibrated to 0, and you calculate 0, which is wrong. So we decided we need another method for the calibration of these types of sensor. And this is the setup which I built up. We have a broadband light source, quartz tungsten halogen lamp in a monochromator. And this monochromator splits up the light in 5 nanometer steps. We swipe through the whole spectrum of interest. 50% go to a reference sensor where we get watts per square meter and wavelength. And then we can, with the energy, we can calculate the power. And the other 50% of the light go to our multispectral sensor. And what we get from this data acquisition in our setup, we get a reading all 5 nanometers. And to make this clear, it's now here in 10 nanometer for better visibility. So channel F6 decreases, channel F7 increases. You can also see it here in the numbers. Channel F7 decreases and channel F8 comes in. We have a reading of all our channels. We have from our reference sensor, we know the respective power. And what we now need is a kind of regression scheme to calculate our coefficient. So, and you see these channels, they are very correlating to each other. And therefore we decided for a partial least square regression because it is known for to minimize the cross correlation between channels. And with this, we calculated our coefficients. And this is now pretty nice. We have now a small formula. It's 10 multiplications, 9 additions. You can run it on every microcontroller, very cheap, very fast. There's no machine learning. And what we get out of it is this kind, in blue, this kind of quantum response curve. We have here, we are lower responsive to blue photons and higher responsive to red photons. Of course, we are here a bit wavy on top, but it doesn't matter since we are interested in the area under the curve. So it averages out. And to validate our calibration, we did a test outside on the campus of University Freiburg. And you can see really clearly that our multispectral sensors, which are placed here next to a commercial broadband sensor, work out pretty well. So they're very nice aligned. You see There are some deviations in the morning. It's because of this moving shadow here. But in the end, we see that it works, and therefore we also decided to use this kind of sensor in our EcoSense toolbox, where we research directly on the leaf of trees. We have this season, so starting in May until now, we have 84 of this gas measuring sampling cuvettes outside where the sensor is placed on top, and we have 24 fluorescent sensors in our research forest, and they deliver nice results. Yeah, right. Yeah, so what's next? So we could with our multispectral sensor, we could follow the quantum response curve pretty nice. This is very compatible to commercial sensors. And the nice thing about this multispectral approach is we're not constrained to this kind of response. We can weight each channel separately. So we are now also able to, if someone is not interested in the number of photons, but in the energy in this area, we can have just a variable target, where because we weight each channel separately. And on the other side, what we also did is we used the same calibration scheme to not calibrate for the whole band, just for the small bands from 400 to 700, 5 to 6, 4 to 500, and we can now also distinguish the colors. So we can measure on the leaf of a tree not only what's the whole amount of light which drives the photosynthesis, we can also distinguish between the colors. And with this calibration method, we are now able to calibrate for spectral and absolute information. And I think this is also interesting for other people and other applications where you also need the spectral and absolute information. And if you're now interested in our work, please follow our project on GitHub. There's the raw data and the code available for you. And if you're interested in the work of our project, what we do in the forest, how we dig in the soil and climb on trees, then follow our Instagram. Yeah, thank you very much.